Good morning. This is Dr. Amy Goldstein. I am the Clinical Director of the Mitochondrial Medicine Frontier Program here at CHOP. I'm an Associate Professor of Clinical Pediatrics at the University of Pennsylvania Perelman School of Medicine, and it's my pleasure today to speak to you about Clinical Trials Update. This is the last talk for the morning session prior to lunch, and you've heard about the clinical manifestations about mitochondrial disease, some of the therapeutics, and some of the genetic background and studies that we're doing here at CHOP. And again, it's my pleasure to talk to you about the clinical trials. So the problem currently is that there are no FDA-approved therapies for mitochondrial disease. As you have heard, these are complex, heterogeneous, inherited disorders that can present at any age. On average, patients have 16 symptoms that are multisystemic across organ systems. These disorders can present in children or in adults and can span across the entire life of the patient and change over time. There are at least 1,500 genes that are needed to make proteins for mitochondrial function, and of these, about 400 have now been linked to human disease, and this number keeps growing. So when we talk about mitochondrial disease, this is really 1,000 different individual genetic disorders. We know that early diagnosis is very important for early intervention, and for sure, if we have an FDA-approved therapy, we want to intervene as soon as possible. However, we're hampered by a lack of natural history data and a lack of biomarkers for our clinical trials. The solution is understanding disease mechanisms through robust preclinical models, such as the work being done by Dr. Marnie Falk here at CHOP. This means looking at animal models or looking at patient skin fibroblasts to test different therapies. We also want to look to the past for improvements in clinical trial design. There have been over 1,000 clinical trials done for mitochondrial disease, and yet we still do not have an approved therapy here in the United States by our FDA. This is because many of the previous clinical trials lumped all of the patients together, no matter what genetic disorder they had or what symptoms they had. And many of the trials included less than 10 patients. And they did not know what outcomes to follow or what biomarkers to use to measure success with that drug. We want to engage our patients and our families and the patient support groups so that we can prioritize symptoms and goals for clinical trials and find out what would be a meaningful outcome for a patient. This also involves communication with industry, such as the drug companies, as well as the FDA, to make sure that our clinical trials are designed properly. Again, I cannot emphasize enough that we need to use the correct outcome measures to show treatment effect. And if there is no outcome measure to measure that particular symptom, then we need to invent one. And a lot of the work that's being done at CHOP now has to do with looking at new laboratory tests to follow biomarkers or to develop new measurements for muscle activity. And hopefully you're getting a taste of that today. It's also very important to maximize the patient's health prior to a clinical trial using the vitamins and supplements as part of the MITO cocktail, especially ones that are responsive to disease, such as riboflavin, coenzyme Q10, and to correct deficiencies such as vitamin D deficiency or iron deficiency. We also want to maximize our patients' nutrition as well as their mobility. So what are the steps for clinical trial design? Again, we need a natural history study so that we can understand what would the disease look like without any intervention, so we know if a drug is actually having a positive effect on the outcomes. This will also include partnering with our patient advocacy groups, the involvement of patient registries to understand the age of onset, the evolution of symptoms, the morbidity and mortality, and any genotype-phenotype correlations, meaning within the same disease, the specific genetic variant may turn out to have a different phenotype or different symptoms. And there are many examples of that in several of our mitochondrial diseases. Again, we want to select the proper and optimal outcome measures. These need to be robust, sensitive, and specific, validated for mitochondrial disease, and clinically meaningful. And the FDA has really emphasized that we need to be measuring how a patient feels, functions, and survives. This may require the use of composite scales, quality of life scales, and biomarkers. Finally, for clinical trial design, we want to find out what are the barriers for participation in trials, and this might include cost, distance from a center, the burden of having to go in frequently and undergo testing. And it will also require multi-center collaboration because as a whole, these disorders are about 1 in 4,000 people, but some of these disorders individually may be less than 100 people affected. So it's going to take a big partnership worldwide. There are many therapeutic targets, and I'm going to start talking about the current clinical trials and some of our recently completed trials. And this is a slide that shows you the nucleus of the cell in the bottom left, the cytosol or the cytoplasm, and then the mitochondria on the upper right-hand side. And as you can see, there are many pathways that involve the nucleus. Some of these, starting at the bottom left, involve the sirtuin pathway, PGC1 alpha, and the PPARs. And this is very important group of drugs that can affect this pathway. And these are some of our nutrient sensing pathways and mitophagy pathways. And I'll talk about this in a little bit later slides. I'm going to talk a little bit about gene therapy using these viral vectors to insert genes. And then the effect on the mitochondria, we're going to talk about ways to intervene with mitochondrial DNA replication, giving nucleosides. So these are little parts of the mitochondrial DNA. The use of L-amiprotide, which just finished its trial this past January to stabilize cardiolipin on this intermitochondrial membrane. Some of the antioxidants that have been used that will help reduce reactive oxygen species and replace glutathione. And then therefore help with DNA damage and preventing lipid peroxidation. So these are just some of the many points of intervention in the cell. There's a long list of clinical trials that are ongoing, and these are available at clinicaltrials.gov. And some of these have been in trials for several years. Some of these are brand new, and I'm going to go through them by category. But again, you can keep up to date with clinical trials by looking at clinicaltrials.gov and putting in the word mitochondria. I also want to show that currently in the clinical trial development pipeline, there are many phases of where we are with some of these drugs. Some of these are still in vitro models. Some of them are in vivo models, meaning in an animal model. And then there are several phases of drug trials, including phase 1, phase 2, and phase 3. And finally, resulting in regulatory approval. You'll see that right now only one has been approved by the European Union, and that is a drug called idebenone, which is one of the antioxidants, and it's been approved for use in Leber's hereditary optic neuropathy. It is not currently FDA approved. And again, there are no FDA approved therapies at this current time. I'm going to talk about these again in terms of antioxidants, targeting mitochondrial biogenesis through certain drugs, membrane stabilizers, targets of mitophagy, enzyme bypass, gene therapy, and then some miscellaneous trials. So first we start with antioxidants. Currently, we use several antioxidants. Coenzyme Q10 is one of the antioxidants that we use in our mitochondrial cocktail. Idebenone, as I just mentioned, has been approved by the European Commission for Leber's hereditary optic neuropathy. It's also been in trials for Friedreich's ataxia. Epi-743 has been developed by a company that was called Edison Pharmaceuticals, then BioElectron. Now they're called PTC. And this is a drug that has been in trials for Lee syndrome, and it will be re-entering trials soon for specifically mitochondrial epilepsy. There's also a drug that has been developed in Nijmegen, Netherlands. It's been in trials for MELAS currently, and this is known as KH176 or sonlicromanol. And this again is another antioxidant to help reduce oxidative stress of the mitochondria. There are several agents that can stimulate mitochondrial biogenesis, literally growing more mitochondria. And these include bezafibrate, resveratrol, omaveloxolone, which is an NRF2 agonist. And just to say about omaveloxolone, this has now been published. This was the Reata drug that unfortunately failed to meet its endpoints clinically for adults with mitochondrial myopathy. And some of the studies that were done during this trial included a 6-minute walk, exercise testing, and muscle biopsy. There's a drug called REN-001, which is a PPAR delta agonist. And again, this is part of the nutrient sensing pathway that tells the mitochondria when to replicate so that we can grow more for energy demand. There's a drug called ICAR that has been used in preclinical models. And then finally, we have the NAD plus derivatives. So that includes niacin, niacinamide, nicotinamide riboside, and KL1333. And some of these have been used in clinical trials, notably the nicotinamide riboside, with some variable results in terms of its effect on the muscle and its effect on the brain. So again, none of these are approved at this current time. Theoretically, they certainly sound like a good therapeutic option, and more to come on these medications. Cardiolipin stabilization on the inner mitochondrial membrane. The drug name is elamiprotide. This had been called SS31, then MTP131, then Bendavia. It is now known as elamiprotide. This just failed its primary clinical endpoints in primary mitochondrial myopathy. And the drug was removed from the patient population using it in the trial as of January 2020. However, it is still in trial for Barth syndrome. And there are still data to be looked at for Leber's hereditary optic neuropathy. For Barth syndrome, which is actually a deficiency of cardiolipin, there may be some promising results and more to come in terms of hearing about this drug for Barth syndrome. Mitophagy is the removal of damaged mitochondria, and this is drugs that utilize the mTOR pathway. mTOR stands for the mammalian target of rapamycin. There's a drug known as rapamycin that is an immunosuppressant. It's used in the transplant world, but this was noticed in preclinical models to help survival of mitochondrial model in a mouse. And it has now been used in humans off-label, and there's a lot of work being done to see if we can bring this for clinical trial. There are several derivatives of rapamycin, including everolimus and then this nab-sirolimus, ABI-009. And this is the one that currently is listed on clinicaltrials.gov, and again, hopefully will be coming soon for a clinical trial so that we can see if this is going to be helpful for mitochondrial disease. Enzyme bypass has been thought about in preclinical models, and I'm not going to spend a lot of time. Hypoxia has been seen to be beneficial in a mouse model of mitochondrial disease, and at this current time we are not recommending that our patients are hypoxic, but we do think that over-oxygenation or hyperoxia is not good for our patients with mitochondrial disease. Therefore, the use of supplemental oxygen, if there's not an oxygen deficiency, is not recommended at this time. Too much oxygen can also develop oxygen-free radicals that can cause more cellular damage. Nucleoside replacement therapy, I mentioned in one of the cartoons. This is right now in clinical trials for a disease called TK2 or thymidine kinase 2. And this is using nucleosides replacing the pyrimidines, so the C and the T in the DNA. And the results are remarkable at this time for the open label study. And there's currently a clinical trial for TK2. And Columbia University in New York is the big center for this disorder. It is also being proposed to look at the other mitochondrial depletion syndromes such as POLG, DGUOK, and currently there's a lot of question about whether this therapy would work for those other disorders, and caution is needed at this time. Enzyme replacement therapy is in the works for thymidine phosphorylase for a disease called MINGI, mitochondrial neurogastrointestinal encephalomyopathy, either as platelet transfusion or as a bone marrow transplant, or in its new form as erythrocyte encapsulation. So more to come for enzyme replacement, and this may be, again, another possibility for other mitochondrial disorders in the future. Solid organ transplant has been used in several of the mitochondrial disorders. And again, this is a model for replacing some of those enzymes, but it really does not pan out as well as it does for other inborn errors of metabolism. And of course, we worry mainly about the brain and the ability for the brain and the nervous system to be aided by some of these enzymes for some of these disorders. And of course, if there is liver failure, kidney failure, or heart failure in some of these disorders, then solid organ transplant should be considered in these patients, but not as a means for enzyme replacement. Finally, I want to get to gene therapy. Everyone asks about gene therapy, and there are viral vectors that are approved for use in other neurologic disorders. And again, you insert the gene that's necessary into the AAV vector and then give it to the patient. And this is being used for Hunter syndrome, Sanfilippo, Gaucher, GM1, gangliosidosis, Pompe disease, and finally spinal muscular atrophy. Gene therapy is currently in clinical trials for Leber's hereditary optic neuropathy. There are 2 trials, and I believe they are both now closed with the data being examined. One is a direct viral vector for LHON, and then the second one is a recombinant viral vector, and this is very interesting. This is actually using the protein that's missing with a mitochondrial targeting sequence, and so the actual viral vector is going into the nucleus where the protein is then made, and then the mitochondrial targeting sequence added to ND4. So this is a gene that should have been made by the mitochondrial DNA, but there's a mutation there. So it's a very tricky thing using the nucleus of the cell to generate the mitochondrial protein and then have it delivered in the right place. One of the things that's very interesting about this trial is that they injected one eye only and found improvements not only in the eye that was injected, but in the other eye. And so there's a question about whether the gene is actually able to travel through the optic chiasm into the other eye. And again, the data are now being looked at very carefully. Currently, 2 gene therapies hold FDA approval for the treatment of spinal muscular atrophy and retinal dystrophy. This is for Leber's congenital amaurosis, which was discovered here at CHOP and has been very successful. And there are preclinical models for gene therapy right now for Mingy, ethylmalonic encephalopathy, MPV17, and 2 models of complex 1-Lee syndrome. There is another mechanism called MITO-TALENS. TALENS are the mitochondrial transcription activator-like effector nucleases, and this is for mitochondrial DNA. There's a lot in the lay literature about CRISPR-Cas9 technology, and this will not work in the mitochondrial DNA, but MITO-TALENS are nucleases that will selectively find the mutations in mitochondrial DNA and then excise them and literally destroy the mitochondria that have the mutations, leaving the wild type or the normal healthy mitochondrial DNA intact. And so this is, again, still in the preclinical model, but might be a promising future. So thank you very much for your attention. And with that, I'll take any questions and be happy to answer them.